The last um, the last lecture ended up with the idea that there's nutrients continuously moving into fresh waters or marine waters for that matter too, and that it's a very dynamic process, and that standing stocks don't tell the whole story of. So if you measure the amount of ammonia in the water, even if it's low, that doesn't mean that it's not a productive system, because organisms could be taking it out very, very quickly. And even in the most productive systems, ammonium and phosphate levels can be very, very low. They're just turning over very quickly. And the reason for that is nutrient remineralization, or sometimes called mineralization. Um, and this can come from several places. Healthy cells leak some, they're not perfect. They also excrete things like proteins, the enzymes and things like that at times to alter the environment around them. Many animals and bacteria excrete, heterotrophic bacteria excrete, and we'll talk about why they do that to maintain their homeostasis and balance of their stoichiometry. We'll talk more about viruses, and we've talked about them some, but they lyse cells and they reproduce and they can release nutrients. And then when we get to the microbial loop, we'll sort of discuss in more detail how mineralization is dominated by microbes. And then the final one is that things like migrating salmon and hippopotami and beavers and migrating zooplankton can move materials from one place to another and then excrete and have it be important. One of the things when we talked about Hutchinson's paradox of the plankton was that lakes are well mixed. And as people considered this, they really started realizing that maybe they're not. And the reason for that is that there are zooplankton that are excreting, and there are algae and maybe bacteria that are taking up nutrients, and they're doing so at very small scales. So diffusion should be very rapid, but there are processes that are pulsing nutrients. And one of the things that led to looking at this is the idea that we have large algal cells that are poor competitor for nutrients because they have a low surface area to volume ratio, right? They can't pack as many, let's say, phosphate uptake proteins on their membranes if they have relatively small amount of surface area to the entire volume of that cell. And even though that's the case, we oftentimes, even in systems where there doesn't appear to be a lot of grazing pressure that would, that largest cells would be resistant to, even so, there's oftentimes large cells that shouldn't be great competitors. And one of the ideas is that pulses of nutrients may last long enough to give large cells an advantage if they run into the patches. So because they have a big volume, if they can take it up really quickly at very high concentrations. Remember, we had the Vmax, right? The Michaelis-Menten uptake curve, and uptake increases up to a point. Well, the large cells can pack away more and continue to take it up and store it better. So are these patches stable? And so there's an experiment done by John Lehman and Don Scavia, which got at that, which I think is kind of elegant, and that's why I like to talk about it. And they took radioactive phosphate, and put it in an algal culture so that the algae built their cells with that phosphorus. So the algae were radioactive. And then they fed that radioactive food to Daphnia and labeled the Daphnia up. Then they put them in clean place, so they're away from those radioactive algae, right? And let them kind of have it be part of their body. The next step was they took the Daphnia and put them in with unlabeled algae, just in beaker or something like that. And they didn't leave them in for very long. And they filtered the algae out after that onto a filter. And then they put a photographic emulsion on that filter. And the radioactivity of the phosphorus would expose the photographic emulsion. This is just how Marie Curie discovered how radioactivity was discovered. That somebody put a sample of radioactive thing on top of film and it exposed it, right? So that those energetic particles can expose film. So they could look at the tracks and go back to individual cells and see how much label each individual cell had. And what they found out is that some of those algal cells had way, way more nutrient than they should by chance, and others had way, way less than they should by chance. So it wasn't just an average distribution. So that meant that everything wasn't mixed in that beaker, that those pulses that Daphne was putting out when it excreted were taken advantage of by the cells that were nearby, and they packed a bunch away, and the ones that were far away, unlucky ones, didn't get much. So that suggests that pulses of nutrients are important. So not only is mineralization important, but it's also not a homogeneous process. It is heterogeneous. So that's another explanation for the paradox of the plankton, which we talked about earlier. I just thought that was kind of a cool experiment. I've always have since I read it the very first time. So the next thing is moving stoichiometry, and we talked about nutrient, how we can harvest algal cells, and if they're not at balanced growth, that we can see what nutrient is limiting. But we can also think about how stoichiometry of the things that eat the organic material that the primary producers create changes the what they excrete and what becomes available. And anytime you have more nutrient than you need, you excrete it. So we're excreting urea and phosphate all the time, because in our food in general, we tend to get more than we need of carbon. So if we assume the 10% rule of that that animals respire, so they need carbon not only for growth but also for energy, and that growth is at Redfield ratio. The Redfield ratio is 106 to 16 to 1. So then, very roughly, a heterotroph would need food. They'd need 1,000 carbons for every 16 nitrogens and 1 phosphorus. Because if its food is at the Redfield ratio, 106 carbon, 16 molecules of nitrogen, 1 molecule of phosphorus. So if it eats food lower than carbon, if this is 500 or 300 or 100, it's going to be having to eat the food to get the carbon, not the nitrogen or the phosphorus. So it's going to excrete nitrogen and phosphorus. So you can figure out what an organism is going to be excreting based on what kind of food it's eating and what the growth requirements of its body are. So here's an experiment that shows that for bacteria. And they used a variety of different amino acids to feed bacteria. And different amino acids have different carbon to nitrogen ratios. Some of them are very carbon rich and some of them are very nitrogen rich. So the ones on this side are richer in carbon and the ones on this side are relatively richer in nitrogen. When they're very rich in carbon, the bacteria that take up those amino acids retain almost all of the nitrogen, right? Because they're limited by carbon, by nitrogen, excuse me. When they're super rich in nitrogen, then the bacteria are going to be limited by carbon, and they're going to dump out 70% of what they take in. Lane asked, are there any habitats that have carbon as a limiting nutrient? Yes, probably. Anytime when we have heavy nutrient loading into like say eutrophic lake, right? Nitrogen and phosphorus the food's going to be super nitrogen and phosphorus rich, and so in that case, many of the organisms would be limited by carbon. Also, when you move up the food web, a lot of times organisms will be limited by carbon, not by nitrogen or phosphorus. Like us, for example, we eat up the food web. And that's because if you think about our food is at roughly at, our food is roughly at Redfield ratio. Now, if people are eating all carbohydrates and have a very low protein diet, then they'll be nitrogen limited, and they'll be, they'll have poor amino acid. They won't have enough. So it's possible to be limited by other things, and same with animals. But there are habitats that have carbon as limiting nutrient. That's a good question, Lane. And different animals vary in their stoichiometry. And so this is why I put the copepods, the Daphne up, because I'm going to be talking about Daphne and copepods. And so we see that a Daphne in general tends to be richer in phosphorus than nitrogen relative to a copepod, which tends to be richer in nitrogen than phosphorus. Okay, so you can guess which of these animals is more likely to be limited by phosphorus in their diet, Daphne or copepods. Okay, so Daphne is more rich in phosphorus, copepods are less rich in phosphorus, which is most likely to be limited by the amount of phosphorus. If there's the same amount of phosphorus in the environment, which one's more likely to be limited by phosphorus? Good, Mac, Daphnia, Abigail got it, Daphnia, and Cassie got it with question mark. Yeah, that's right. Daphnia needs more phosphorus, right? So it's going to be more likely to be limited by it. Copepods are going to be more likely to be limited by nitrogen. So given food that's sort of semi-balanced, right, if there's a lot of Daphnia, they'll be excreting a lot of nitrogen and keeping more of the phosphorus. If there's a lot of copepods, they'll be excreting phosphorus and keeping more of the nitrogen. So they can alter the nutrient limitation of the system because they're the mineralization that's putting it back in the system. So let's just give an example here. We have the nutrients in food. So now what I do is I've got some algal cells that have 1,000 carbons and 4 nitrogens and 1 phosphorus. So the first thing, if they've got 1,000 carbons, why would they have 1,000 carbon? And what might lead to having a bunch of, instead of being at 106 to 16 to 1, then what would lead to that? Well, they can make food, right? So let's say it's a photosynthetic organism and it's an alga, right? So it's got a lot of carbon. Yeah, they could be making a lot of cellulose. So carbon's probably not limiting that organism, right? It's probably nitrogen or phosphorus. And which one is probably more limiting of those to the algal cell here, nitrogen or phosphorus? So first carbon, then secondarily, there's one that's maybe more limiting. Lane's wrong. Right. Sean, Trevor got it. Sorry, Lane. Good guess, though. Right, because this is supposed to be 16 to 1, but it's only 4 to 1, right? And so if there's less nitrogen there than there's supposed to be to build a full cell, they're obviously hurting for nitrogen. Okay, so that's the first step. Now we've got this copepod, and it's eating this food, right? And it's eating food that's 1,000 carbons, 4 nitrogen, and 1 phosphorus. What is limiting? What's the limiting nutrient in this case? Right, so you can sum these two. Right, good, Trevor, good. Morgan, we got a couple saying nitrogen, right? So you sum these two and they come up to 1,000, so that, and the phosphorus is right there, but nitrogen's low, right, relative to what it needs to build its cells. So it's going to be nitrogen limiting. So if it's nitrogen's limiting, what is going to be excreted in this case? Right, phosphorus, and maybe something else. So how many of these is it going to need to eat to get to the 16 nitrogens? Good, right, yeah, you have to eat 4 of those, right? So if you eat 4 of those, how many phosphorus is it going to be getting for that? Be getting 4, right? And how many carbons is it going to get for each of those? So by the time it gets 16 of these? Great, Abigail, 4,000, right? So it's getting 4,000 carbons, 4 phosphoruses for every 16 nitrogen. So it's going to have to be dumping carbon and dumping phosphorus to bring in as much nitrogen as it needs to grow. All right, so in this case, it's going to be mineralizing phosphorus faster than it's mineralizing nitrogen. It may excrete the carbons either after it takes it into its body, or it could just not eat it, just not assimilate it. Now, one of the things that cells can do in reality is they can pick and, animals can do in reality is they can pick and choose their food. So we know, for example, that bison will eat newly burned grass because it has higher nitrogen content, and they're dumping the carbon out, and they're trying to get the nitrogen, right? It's the same thing with animals that are in that are grazing in lakes and streams. Okay, good. If for some reason you're not answering these questions, you got to answer them, because I won't have your question of the day points. Okay, so the next thing to do is look at carbon to phosphorus ratios. And we see that that varies by different types of organisms, right? And we see that fish have the highest amount of phosphorus relative to carbon. And then as we move down, and the mollusks have their shells taken off, by the way. This doesn't include the carbonate shell. And so the insects are the most phosphorus poor, and the fish are the most phosphorus rich. What do you think it is about fishes that varies from these other organisms that means they have such a high phosphorus demand in their bodies? Anybody tell me how fishes vary from the other organisms that are on this list? Not gills, no. Gills don't need much phosphorus. They're just tissue. They're they have about Redfield ratio tissue. And I'll give you a little hint. Yeah, Cassie got it right. So what's the major group that all these are part of versus the fish? Right. So these are the fishes, as Cassie said, a vertebrate, right, Trevor? These are all invertebrates, right? That means what don't they have that fishes have other than vertebrae? In addition to vertebrae, or vertebrae are part of it. Bones, right? Bones are phosphorus rich. They're made of apatite, among other things. They have a high phosphorus demand. So we have a relatively high phosphorus demand because we have a lot of bones relative to a lot of invertebrates. So that means that there's a little bit of difference in what the requirements are. And then in addition, they went through, in this case, they looked at a bunch of these are stream organisms, predators, grazers, and detritivores, and they saw that the C to P ratios fell like this. Well, most of the predators were fishes, right? Many of them were. I mean, there's stoneflies and dragonflies and things like that are predators as well. And then some of the grazers are fishes and some aren't. So one of the things that happens is that these predators are getting relatively phosphorus-rich food, right? Because they're eating other animals, and other animals tend to be phosphorus-rich relative to, say, algae or something like that. So they can get by with more phosphorus. They can get more phosphorus in their diet. Things that are eating detritus, that can make it on detritus, are just eating, leaves and stuff, and it's pretty nutrient poor stuff, right? There's a lot of carbon in it. So they're closer matched to their food than, and so they're closely matched to their food, so they have a higher carbon to phosphorus ratio. So this is sort of what we're talking about, is the field of ecological stoichiometry. And it's a whole subfield of ecology that basically controls some nutrient cycling in the way that food webs work. And that's why I have been covering it. And here's a practical example of that. And this is Michelle Evans White, who is in my lab as an undergraduate, and then a graduate student, and then she came back as a postdoc, and now she's a professor at Arkansas. But she took, I think, the second time this class was offered, she took it from me. In any case, these are data from Nebraska, Kansas, and Missouri on streams, and the collection is phosphorus. Total phosphorus is on the x-axis, so that is the independent variable. And the dependent variable is the diversity of invertebrates, stream invertebrates. And what we see is at low phosphorus concentrations, the diversity is the highest. So this is low amounts of phosphorus pollution. And it goes up, it drops for a while, and then it kind of levels off until we get up to a high enough phosphorus. What Michelle hypothesized when we saw this was that if you have invertebrates that have a high growth rate, they have a high phosphorus demand. And that's because they need a lot more DNA and RNA and phosphorus, and mainly to keep things going right. So they have a high phosphorus demand. They tend to have a relatively low carbon to phosphorus ratio. These rapidly growing species can outcompete other species. However, if there's not enough phosphorus, they can't grow rapidly. So when you have high phosphorus, the things that specialize on high phosphorus can dominate everybody and knock out the rest. When you have low phosphorus, then there can be more diversity because some things can specialize on high phosphorus and pick it out, and others can eat poor quality food and get more get their phosphorus that way. Then to test that hypothesis, you said, well, if their predatory species are eating other animals, and those are relatively phosphorus rich, so those other animals should not be as dependent upon the phosphorus in the environment, and diversity should not be as dependent. And she was correct there. So while this is still a correlation, there's less of a heavy dependence on the phosphorus, on diversity on the phosphorus in the system. So this is an example of now we can use invertebrate diversity, right, to get at the degree of phosphorus pollution in the system. And even more so, this suggests that in this range of phosphorus is where organisms naturally evolved, right? They're tuned into the range of phosphorus concentrations. This is not as normal of an environment because there's fewer organisms that are evolved to tune in to being in that kind of environment. And they haven't partitioned the environment as well. So there's a practical example related to stream eutrophication that will tie into the next chapter that we talk about.